So chapter 5 is about the function of cells. Chapter 4 was about the structure of cells. And so in chapter 5, we're going to actually talk about three kind of distinct topics. So the first one is the membrane structure and function, and that we're talking about the plasma membrane, but all membranes throughout the eukaryotic cell. Then we're going to talk about energy in the cell, and we're going to talk about the molecule of ATP. And then the third part is about how enzymes function. So enzymes are a good example of our default answer in this course, that they are proteins. And we're going to talk a little bit about how enzymes work, because the next bit that we go into are some chemical reactions that happen in the cell, and we need to understand a little bit more about ATP, the molecule of energy, and enzyme mediated reactions. So kicking it off, the idea of the membranes in a cell. The membranes, if we were to roll in what we learned back in chapter 3 with macromolecules, are all made of phospholipids. So we're going to really focus on the plasma membrane, the outer cell membrane, but technically all membranes are made of these phospholipids. Now, the job, the function of the plasma membrane is to control what goes in and what goes out of the cell. And we call that selective permeability, not semi-permeability, but selective. It controls what goes in and what goes out. There's a few buzzwords or kind of key terms in here, and you'll notice that they're in red in my videos. Your handouts are in black and white, maybe if you got the printed version. So yeah, it might be nice to just really focus on a few keywords that can anchor your understanding of membranes in the cell. So the structure of membranes is really what allows this job to happen, that theme of structure and function. So the structure of these are they're made up of molecules, phospholipids, like I'd said, and they're a two-layer sheet. So we call that the phospholipid bilayer. This is sort of the next key piece of vocabulary that explains what we're talking about with structure and function of membrane. So the phospholipid bilayer we've seen before when we were introduced to phospholipids, they have the hydrophilic or the water-loving head. So water can be out here, and the hydrophobic tails. And they're sandwiched here in the middle. So these tails would be hydrophobic. And then on the outside again, we have the aqueous environment of the cytoplasm of the cell. So this diagram over here, I didn't have enough room to label it, but this idea, the hydrophilic heads are exposed to the water and the hydrophobic tails are shielded from the water. You could write that out, or you could label a diagram quite clearly with that. Okay, so we've got two key words. We've got selective permeability, or key terms, I guess. They're more than two words. Selective permeability, phospholipid bilayer. And then the third one would be sometimes membranes are described as a fluid mosaic. So there's two words in that, and each one has some kind of a clue as to the other macromolecules that are involved in the structure of a cell membrane. So a mosaic means that there's more than one component to this. So like if you make a tile mosaic, you've got lots of different tiles. In this, we have a lot of different molecules. And the key one that makes it a mosaic structure would be proteins. And the proteins have a lot of key jobs that are going to be on the next slide. So see next on that one. And then the other word here, this idea of fluidity, that has to do with an important lipid molecule, cholesterol. And that basically kind of greases the gears. It allows these molecules to kind of freely flow around each other and not be locked into place. So here we have that phospholipid bilayer, and now we have the proteins that are embedded in it. And so these proteins have lots of different jobs. And if we take a look at their key jobs, I mean, there's lots of different jobs. I'm going to focus on a few that really are ones that we've talked about before. So the first one would be enzymatic activity. So proteins are enzymes often, or enzymes are often proteins, I should say, and even in the plasma membrane or even in cell membranes, they hold that role as um, making chemical reactions happen. The last topic in this chapter is enzyme activity, so we'll expand on that a little bit. The next thing we could talk about is cell-to-cell recognition. And so you have identifiers on your cells, and the most common example of this would be blood typing. So I'm type AB blood. That means I have A markers and B markers. And what I'm talking about are these markers here. Now they're half protein and half carbohydrates. We call that a glycoprotein. But the protein part is still a big deal when we talk about how cells recognize each other and recognize your own cells. If you have type O blood, that means you have no markers on your red blood cells. The third kind that I'm going to kind of zone in on here because it has relevance in stuff that we're going to study, and it's often easier to have context for these things rather than just a big list of rather abstract concepts. So the last job that I'm going to really focus on here is transport work. And we're going to talk a little bit about how things get into and out of the cell. And there's two different kinds of transport work that use that. I'm going to say them now, but they're in the notes coming up. So we have active transport, and we have facilitated diffusion. You may have noticed those from the pre-reading, so I'm just kind of linking them in there now, but that's really the next topic that we're going to discuss. So how things get in or out of the cell, the first kind of processes that we're going to talk about are passive transport. That means that they don't use energy. We're going to, throughout this lesson, be able to use those two words somewhat interchangeably. We'll probably shift more from energy to the molecule of ATP, but ATP is like the currency of energy. It's how energy is packaged in the cell. And so for passive transport, you don't need these. So all of these can happen without any energy. And oftentimes when I talk about this, I talk about, getting firewood. And so if you want to get firewood from up above the road, that's going to fall down without too much energy. If you are going to get firewood from below the road, that means you're going to have to add a whole lot of energy into that. So coming down the hill or down the gradient, this would be passive. And coming up the gradient or up the hill, this would be active. So focusing on these passive transports first, we have a few different kinds. The first one is diffusion, sometimes called simple diffusion. So if we have things dissolved in our fluid, in our cytoplasm, or in the solution that a cell is in, things are going to move from where there's lots to where there's little. We want to go from high to low concentration. This would be down the concentration gradient. Things flow downhill. That's pretty easy, right? And so an example of this, not as far as cell transport goes, but could be when somebody opens a container of leftovers in the lunchroom and the smell diffuses across the room. So same thing goes, if a cell is in a solution that has a lot of salt in it, that salt is going to diffuse into the cell. It moves from where there's lots of salt to where there's a little bit of salt. The next one that I'm going to talk about is facilitated diffusion. Now, it's mostly the same as diffusion, but it has a helper. And as you might guess, that helper is a protein. And so it still happens downhill. It still happens from high concentration to low concentration, happens down the concentration gradient. But there's a bit of a gate here, and it's a gated protein that allows the substances to diffuse in or out of the cell. So it just depends, what's on either side for those dissolved substances to diffuse through facilitated diffusion. So the last one is a special case of diffusion. So this is diffusion of water. So oftentimes water is is what we've got things dissolved in. And so water moves oppositely to those dissolved things. So if we have this cell that's in a really salty solution, like I talked about in number 1, the salt is going to diffuse in, but the water is going to move out via osmosis. So the water moves out because it wants to equalize the concentration on either side of the cell. So the water's going to move to where there's more things dissolved. It's going to move to where it's more concentrated. So oftentimes we have the two working together. And so, this would be the salt diffusing in and the water moving via osmosis out for the same reasons, this idea of a concentration gradient. And again, all of these happen in a passive process without any energy expended by the cell. If we were to talk about, the solution that something is in, and I've already talked about a hypertonic solution. What we're talking about is the tonicity. So tonicity is a term that describes the ability of a solution to make a cell gain or lose water. So tonicity is dependent on the concentration of things on either side of a membrane. So if we talk about a solution that is isotonic to the cell, that means that it has the same concentration on both sides. That means that water might be moving in and water might be moving out, but there's no net movement of water. Your eyes might dart to the next slide because there's some images that I find quite useful because it has diagrams of these. I'll talk through them as we go through that next slide, but I do find that to be a useful thing. The second one would be a supercharged concentration. So, as a supercharged situation could be, that'd be a lot of salt outside the cell. And so, like I said, water or salt is going to move in via osmosis, but the key thing with tonicity is that water is going to move out. And that's going to have an effect on the cell, as we see on the next slide. So the third one would be a hypotonic solution. And so that means that inside the cell, there's a lot of dissolved things. So what's going to happen here as far as water, right? That's the key thing with tonicity. Water is going to move in. Yeah, the salt is also going to diffuse out, but with tonicity, we're more concerned about the movement of water. And so the water moves in because it's trying to equalize those concentrations. There's lots of dissolved stuff inside the cell. There's not as much dissolved stuff outside the cell. And so the water moves in to equalize that concentration. I've already talked about a concentration gradient, but that's what I was talking about, things moving downhill, right? The gradient is really just that difference from inside the cell to outside the cell. So things diffuse down the concentration gradient and the water moves oppositely to equalize those concentrations. So here are the pictures. If you were an animal cell, and in class I can't help but sort of act this out, so you're kind of missing something by not seeing me in person right now. So the first one is isotonic. That's what it said, where it's at equal amounts, so there's no net movement of water. Water's always moving in and water's always moving out, but in equal quantities. So these would be normal. They would just be a normal plant cells. The hypotonic and the hypertonic, there's a really significant effect on the cell. So animal cells at the top have just the plasma membrane. So the hypotonic solution, as we have a lot of things dissolved, in the cell, got this almost backwards, right? The water's going to move to dilute that. Excuse me. And when the water moves in, it's going to cause the cell to swell or potentially burst. In a plant cell, it has both a cell wall and a cell membrane within that. And so as the water moves in, that cell wall really fortifies the structure. And instead of it bursting, it's just going to get really plump or turgid or perky or crisp. If you ever pick lettuce in the garden, you might soak it in a sink of water. that water is hypotonic as compared to the lettuce cells. And so the water moves in and your lettuce stays nice and crisp when you make your salad for dinner. The last one here, the hypertonic, that means that there's lots of stuff dissolved outside the cell, and so the water moves out to equalize those concentrations. That's going to essentially dry up the cell and result in a shriveled appearance of an animal cell. For the plant cell, again, it has both the cell wall and the plasma membrane. So when the water comes out, the plasma membrane can shrink away from that cell wall and sort of, still have that structure, that cellulose in the cell wall, but the plasma membrane does shrink away, which is equivalent to that shriveled. The fancy word for this is plasmolized, or it does say shriveled. They mean the same thing. So those are the passive methods, really understanding diffusion and osmosis and tonicity in that. And then also remembering that facilitated diffusion is like diffusion but with a protein helper. There's a link here with the active methods. So active methods, all of these are opposite to the passive transport where they need energy. They need ATP. Things are going to go uphill or against the concentration gradient. This active transport, the sodium-potassium pump, is essential in the human nervous system. In order for a cell to fire, it has to put ions of sodium and potassium on either side of a nerve cell membrane. And so it's going to pump up this gradient against its kind of inclination. So it's going to put sodium, it's going to push sodium to where there's already lots of sodium, and it's going to push potassium to where there's already lots of potassium. And so because it's going against the gradient, you need energy to do that. The other two are probably more significant, seeing as we're not studying the human nervous system. So coming up with examples that are relevant in our course, I think is really important. So endocytosis and exocytosis are when the cell membrane takes in materials. And so for endocytosis, it could take in a liquid, and that's called pinocytosis, or it could take in large molecules or parts of cells, which is phagocytosis. Pinocytosis, I don't have a great example. It would just be taking in dissolved substances that it would require, so maybe some ions or something. But phagocytosis, this is key in your immune system. You have little cells in your immune system that work like Pac-Man. And so they literally engulf or eat things that are invading your body. Exocytosis is the opposite of endocytosis, and it doesn't have the fancy words about liquids and solids. So the next page gives a little bit of a picture of this. So phagocytosis would be food or the particle of it coming into a food vacuole. Remember vacuoles for storage. And then pinocytosis is drinking action. And so here we can see dissolved substances coming in as the plasma membrane folds on itself, making a vesicle for transport. This slide can really summarize the key parts of cellular transport.